Okay, very good. So we continue our lectures today in order to finish whatever remaining. We are on the page 37. So after knowing that many microbes are present in the rumen of ruminants, have an important role in the nutrition of ruminants such as cow, now we can have a brief explanation about which types of bacteria and protozoa are there and what are their activities. Bacterial activities in the rumen. Many bacteria produce acetates or acetic acid, predominant acid in the rumen. Among acetic acid, propionic acid, and butyric acid, the most quantity is related to this acid. Many bacteria produce propionic acid, the only fermentation acid that can be converted into carbohydrate by ruminant. Whatever animals like ruminant receive, they go through the catabolism. This acid can be used for catabolism and anabolism also. Some nitrogen fixation activity in rumen is done, but it is very small amount. It is 10 milligram of nitrogen per head of cattle per day. It is very small. It is not significant amounts because ammonia present in the rumen repressed nitrogen fixation. So there are some proteins in the plants, and bacteria by use of those proteins produce ammonia, and ammonia repress nitrogen fixation. So whatever is fixed is very in a small quantity. It is not sufficient amounts because ammonia present in the rumen repress nitrogen fixation. Ammonia present in rumen can converted to microbial protein by microbial community, subsequently digested by the ruminant. So bacteria present in the rumen change cellulose to acids, acids go to blood, by blood going to cells, and in the cells changing to ATP. And by effect on proteins, ammonia produced or absorbed by protozoa for their protein synthesis, and again ruminant can use protozoa as food and their proteins absorbed. So mostly carbon source is absorbed by bacteria and mostly nitrogen source is absorbed by protozoa. Protozoa activities in rumen mostly ciliates, but some flagellates are also present. They grow anaerobically and ferment plant materials for energy and tolerate the presence of dense bacterial population. So protozoa don't feel bad by the presence of large amounts of bacteria because they don't go to competition with bacteria. Bacteria receive cellulose, use it, and protozoa do it so. And on another way, not only cellulose and the other constituents of plants are food for protozoa, all the bacteria present there are their foods. So protozoa have two sources of food. what cow eat, plant materials, and all bacteria present there. So they feel very well. Their foods are ready made and there is full of foods for protozoa. Some digest cellulose and starch. Other ferments dissolve carbohydrates. And some are predators of bacterial population and store large amounts of carbohydrates. On the other hand, so food comes from animal to bacteria, from bacteria to protozoa, and at last, again, animal can digest those protozoa and receive all carbohydrates and proteins by use of their biomass. Digestion of protozoa occurs in omasum and apomasum, two parts compartments of ruminant stomach located adjacent to the rumen. Carbohydrates to bacteria, then to protozoa, so it is a valuable food chain there. Protozoa are probably digested than bacteria. So it is better for animal to digest protozoa because protozoa don't have cell wall like bacteria. The hard work is done by protozoa. So protozoa digest cell wall of bacteria and receive the content of bacteria, then transfer these items to the ruminant. Microorganisms in the rumen digest plant materials, making low molecular weight fatty acids and microbial proteins available for utilization by ruminant. Rumination process. What is rumination process? So when ruminants such as cow are in a field, they are in competition with each other. It is not so the food to be as much as they need. So they go very fast eating without chewing. So the plants come down to the rumen, to the stomach. After winning the competition, then by chance, in suitable time, they return back those plants up to the mouth and again re-chewing. First time, fast chewing only for eating. Second time, chewing for a long time to cut foods as much as foods to be cutted. The effect of microorganisms can be better on the food. So we can see the alimentary canal of one ruminant is look like a biotechnology factory. Here there are a large amounts of workers are working in the alimentary canal to changing the substrate to products which can be used by ruminant cow. Rumination process, rechewing previously ingested food grains, the plant material, and provide increased surface area for microbial attack. Movement of ruminant stomach supplies mixing for optimal microbial growth. and metabolic activity. So some devices which we have in the lab, it is according to the life of ruminants. So we have shaker incubator, yes. When we have flask or tubes, we put in the shaker for aeration and better effect of microbes to the substrates with good temperature. So the stomach and rumen of ruminants are working look like shaker incubator. But if you know, for example, when you have a tube or you have a flask, as much as the microbial population produce acids, alcohols, or another things, they have negative feedback. What is it about rumen? So in rumen, large amounts of acids are produced. Is there any negative feedback? No, there is no negative feedback because all the acids which are produced there are absorbed by blood vessels, and all acids transfer to cells. Those acids going to Krebs cycle, producing NADH2, FADH2, and then going to the electron transport chain in mitochondria, accompanying with ATP production. So the rumen and stomach of cow is look like a shaker incubator. Good mixture of substrates and microbes and good temperature. And on the other hand, it is look like a thermostat or turbidostat, which allow those metabolites, those acids, acid acetic, acid propionic, acid butyric, remove the stomach and rumen going to blood, so new acid production is possible. New acid production is possible. Removal of low molecular weight fatty acids from rumen by absorption to animals' bloodstream allows the continuum of prolific growth of microbial populations. High diversity of microbial populations in the rumen allow them to act on different foods on ruminant diet. So different foods are different plants, okay? The ruminants and cow life is dependent on plant, but different plant like cellulose, chitin, pectin, hemicellulose. So plants containing different types of composition. Accordingly, different types of microbes which can degrade them are present there. There are some another animals which they are not ruminant, but they are ruminant-like. Other animals with ruminant-like digestion are clobid monkeys, sloths, hippopotamuses, camels, macropod, marsupialis. The enzymatic activity on cellulose produce volatile fatty acids on foregut. Even though they are not ruminant, they don't have rumen, but they have a special part with the name of foregut containing these types of microbes, cellulolytic microbes, and changing cellulose to volatile fatty acids, and these fatty acids absorbed by blood and transferred to the cells and are used to form ATP. And some animal herbivores Some animals which their lives are dependent on plants are not ruminant. So what are they do? They don't have any rumen. In non-ruminants, mammals subsist on plant material like horse, pigs, rabbits. Microbial cellulose digestion occurs in an enlarged cecum at the end of large intestine. At the end of large intestine, their cecum in comparison to the other animals are so much bigger. So their cecum in comparison to the cecum of human or cecum of another animals, they have a large cecum with production of volatile fatty acids. So those microbes effective on cellulose are present there, and by production cellulase producing fatty acids, and there those acids are absorbed in the cecum to the blood and transferred to the cells and are used for ATP production. Last part of this lecture is related to the microorganisms in their natural habitats. So if we look to the earth, we can see only three habitats: soil, water, air. We don't have another part. Wherever we see on the earth, we can see water or soil or air. There is no another part. When we talking, so we have atmosphere, lithosphere, and hydrosphere. But when we are talking about those space containing microbes, if we are talking about microbes of soil, we say lithoecosphere. If we are talking about microbes in the water, we are calling them hydroecosphere. If we are talking about microbes in the atmosphere, its name is atmoecosphere. If you didn't forget, so each of them we can consider as habitat, but each habitat containing many niches. For example, upper side of the water, middle side of the water, basement of the water, sediment at the bottom of the water. They are different niches with different situation on that habitat. Okay? Now, for half an hour, we want to focus on atmoecosphere. Atmoecosphere. When we look to soil, we can see some native microorganisms. When we can see to the water, we can find some native microorganisms. Because in the soil and in the water, whatever needed by many microbes are present. They need pH, they need temperature, they need food, they need water. All of them in sufficient amounts are present. But air and atmosphere is not a good place for life, for living, for eating, for growing, for multiplication. So air and atmosphere, doesn't have native microorganisms. Whatever microorganisms we can see in the air, they have come from soil or water. From wind, some soil particles come to the air containing some microbes. By air or striking water to stones and rocks, some small particles of water, aerosols, come up to the air. By coughing and sneezing related to human or animals, many microbes come to the air, but air is not their home. They are not native on the air. Air is only a device for transfer from one place to another place. For example, your place of living is soil. Yes, you live on soil. You don't live in the water and you don't live in the air. If you go to air by airplane, is only for transporting from one place to another place. Air is not our home for living. We are living on soil. So some microbes live in soil, some microbes live in water, but there is no native microbes on the atmosphere because there are not sufficient food for heterotrophs and not sufficient suitable condition for autotrophs. So when we say native microorganisms, they are autochthonous or indigenous microorganisms, which they are permanent members of one place. Microorganisms with high adaptation with physical and chemical environment. So there are many microbes adapted to the soil. There are many microbes adapted to the water. But there is no adaptation to air, because air is not a good place for growing, for multiplying. Allochthonous or foreign microorganisms. If some microbes from soil you transfer to water. So microbes of soil cannot live on the ocean. Microbes of the ocean cannot live in the soil. Microbes of soil and water cannot live in the air. So we call them allochthonous or foreign microorganisms. Transient members such as E. coli survives less than 24 hours when enters marine habitat, but foreign spores survive longer periods. So by sewage of human, if E. coli or coliform transferred to the ocean or sea, after a few hours, they will die because homeostasis of water is not suitable for homeostasis of seawater, salt water is not good for coliforms such as E. coli. So atmosphere, even though contains some microbes, but none of them are native. We don't have any native microorganisms present in the air. Atmosphere consists of three parts. One part is troposphere, from land to 10 kilometers. We call it troposphere. then stratosphere from 10 to 45 kilometers, then ionosphere, which it is more than 45 kilometers. For most parts, chemical and physical parameters of atmosphere don't favor any microbes, don't favor for microbial growth and survival. With increasing height, what happened? For example, if you go mountain climbing, if you go up in the Everest, what's happening? What do you feel? Number one, you feel as much as you go up, the temperature decrease. Top of the troposphere, the first part, troposphere, the top part of troposphere, temperature is minus 43 to minus 83 degrees centigrade. So it is so much cold. It is not suitable for any growth of microbes. So when the top of troposphere is minus 40 degrees centigrade, what can we think about stratosphere and ionosphere? So no microbes can live in ionosphere, not in stratosphere, not even though can be alive on the top of troposphere. Number two, atmospheric pressure. When you go up to the Everest, after some time you feel you cannot breathe. So you need an oxygen capsule because the pressure of oxygen is very low. And oxygen and organic carbon, as much as we go up, decreased. As much as we go up, we understand that different layers of atmosphere filter UV. So as much as we go up, UV radiations are more and more, and this radiation is lethal and has a killing effect on microbes. So these conditions support not only heterotrophic but also autotrophic. So atmosphere is not a good place for growing and multiplication of microbes. Only maybe it can be used for transfer from going from one part to another part. In the second position, stratosphere, along with the previous items, it contains ozone and it is full of ozone concentration. Ozone is another killing agent for microbes. So ionosphere, stratosphere, and the top part of troposphere, never microbes can suffer this situation and they cannot live there. So when we talk about the presence of microbes in the atmosphere, mostly it is related to the lower atmosphere in the troposphere. And lower atmosphere in troposphere works as a habitat and medium for microbial dispersal, only for transfer microbes from one place to another place. Several viral, bacterial, fungal diseases are spread through atmosphere. When one microbe can transfer by air from one infected person to another sensitive person, we can call them airborne infection. We can call them airborne infection. Clouds causes concentration of water. We know that all things can be alive by the presence of water. So if anybody asks what is the most important creation by God is the water. Because whatever life we can see on the earth first is dependent on the water. Because some living things are not dependent on the oxygen. If there is no oxygen, life is there in the earth. Even though we will be killed by the absence of oxygen, but many anaerobic bacteria are present in the earth. But without water, not aerobic, not anaerobic, not in any situation, any microbe cannot be alive. And you can see in astronomy, when scientists searching life on the another planets, first aim finding the water. First aim is the finding the water, because water is responsible for life. So clouds in the sky, clouds in the air possess concentration of water. As well as light is present, as well as carbon dioxide is present. So a little photosynthetic microorganisms can have activity. But when we compare photosynthesis in the air to the photosynthesis in the soil and photosynthesis in the water, it is not comparable. So low amount of photosynthesis in air is not comparable with photosynthesis in the soil or photosynthesis in the ocean. So there are some condensation nuclei maybe supply some mineral nutrients. So for photosynthesis, water, light, carbon dioxide, all are in the atmosphere. And small soil particles in the air are used for what? As the mineral, because photosynthesis needs light, water, carbon dioxide, and minerals. So light is sufficient, carbon dioxide is sufficient, but water and mineral is not sufficient for heavy multiplication like in soil and water. What's about heterotrophs? In industrial areas, there may be even sufficient concentration of organic chemicals that can come to the atmosphere and permit growth of some heterotrophs. But in this case also, the number, diversity, the amount of heterotrophs in the water and in the soil is not comparable with life in the air. So we can say life in the sky is negligible. Many microorganisms from hydrosphere or lithosphere can become airborne, but they are not autochthonous. They can be in the air for short time only for dispersal. Dispersal through atmosphere ensures continued survival of many microbes. Some microorganisms are vegetative, but mostly dormant stage like spores, cysts, radia, such as spores of fungi, algae, some protozoal cysts, some bacterial spores, especially actinomycetes and lichen, inactivate forms of virus can transfer from air from one place to another place. One important types of microbes can easily transported by air are bacterial and fungal spores. And what are their properties which help them to be transported well in the atmosphere? 1. Low metabolic rates. So when their metabolism are low, they need very low water. So for a long time can be float on the air and wait to reach to a suitable place. Large numbers of spores. So when large number of spores coming up to the air, some of them find suitable place and then they grow dramatically. Resistant to desiccation. So they need so much water and they can have those water with them not allowing escaping water from the spore according to the thick cell wall, and they are resistant to UV by help of their pigments. They have a small size, so very easily can float on the air, and low density. With low weight, low density, small size, their floating on the air is easier. So it permits airborne transfer for long periods. Spores are light and they are teeny and light and may contain gas vacuole, helping for floating in the air. They have variety of shapes. Because of their shapes, aerodynamically favorable for airborne travel from one place to another place. Airborne spores. So some microbes can produce spores. By production of spores, those spores can come to air and can travel from one place to another place. Sometimes a spore producer organism help them for going to the air. Sometimes without help, they can go to air. If there is no help of microbe for transfer spores to the air, we call it passive mechanism. Passive mechanism. What is that? Aerosols. What is aerosols? Aerosols such as splash from falling raindrops, rapidly moving water currents striking solid objects such as rocks in river. So small, very small drops of water by striking river or water to stones to rocks come to the air, and by help of wind they go upper and upper. So these microbes are going up These spores are going up, but without any help of mother cell. Second is aerosols, as droplets by mechanisms such as coughing and sneezing by human and animals. Some spores are produced in our body and by coughing and sneezing. So they come to air by our help, not by help of the mother cell. So the second one is also passive mechanism. It is also a passive mechanism. Active mechanism. In active mechanism, the microbe is actively try to send their spores to the atmosphere. For example, there is one fungus with the name of Pilobolus. Pilobolus. Entire spore cluster is ejected when a vacuole in the sporangium base becomes turgid through an increase in osmotic pressure and causes the structure to burst and spores to be carried in a way in a jet of water. So here you can see there is a place for spore production. Spore production is in this part, and inside slowly is full of water, and after some time, by absorbing so many waters, the droplet of water containing spore shoot out like a jet of water. So here the mother cell or the fungus is helping their spores to go far from the mother cell in some distances by help of a jet of the water. In ascomycetes, ascospores are actively discharged to distance ranging from several millimeters to several centimeters. The bursting of ascus is caused by changes in osmotic pressure when glycogen is converted into soluble sugars. Some ascomycetes exhibit phenomenon of puffing, that is simultaneous breaking of large number of asci. So large number of asci you can see are breaking with release of visible cloud of spores. Visible clouds of spores. Here you can see changing the environment conditions such as light, humidity, or temperature can trigger the puffing spores. Sometimes look like here there is a sheet with under drought. This sheet containing the spore contracted in such a way and shoot the spores outside to the air so they can go far from the mother cell. So these items are active form of transfer of spores far from the mother cell. Microorganisms in the atmosphere. By quantitative sampling of microorganisms in the atmosphere by viable plate count, so by serial dilution, In viable plate count, we can count how many bacteria, how many fungi, how many yeasts are present in the air. Direct contact procedure using contact slide. So we can have a slide with sticky substances passed through the atmosphere by an aircraft for fungal spores. We can use a pump, draws air samples in a calibrated volume through a membrane filter with 0.5 micron pore size. Cladosporium is major fungal constituents of atmosphere. Concentration of microorganisms in the atmosphere up to height of 3,000 meters range from 10 to 10,000 per cubic meter. Some microorganisms transported in long distances by air travelers such as Puccinia graminis with rust. So if many things helpful for transferring the spores to the air, what are the factors for removal of microorganisms from atmosphere? They settle due gravitational forces and removing by raining. Why do you like to go out for walking after raining? Because there is a good atmosphere, because many chemicals, many microbes are removed in the air by raining. Okay, it is sufficient. So our lecture over on the page of 43. We don't have anything else to explain. So right now we say that our lecture is over. So next Sunday, yes, our lecture will be followed in next Sunday. We meet each other. We talk about examination. I answer the question and I review whatever you like. Is it any question? Okay, very good. Thank you very much. So if there is not anything else, we can say goodbye and hopefully we meet each other for last class on Sunday. And right now you can leave the lecture and you can leave the class. Thank you very much. Hope the best. See you.